The Tricarboxylic Acid Cycle, also known as the Krebs or Citric Acid Cycle, is a central metabolic pathway that occurs in the mitochondrial matrix. Its primary function is to generate high-energy electron carriers, NADH and FADH2, that fuel ATP production in the electron transport chain. It begins when acetyl-CoA, a 2-carbon molecule derived from carbohydrates, fats, or proteins, combines with oxaloacetate, a 4-carbon molecule, to form a 6-carbon compound called citrate. This step is catalyzed by citrate synthase. Citrate then undergoes isomerization to isocitrate via aconitase. Next, isocitrate is oxidized and decarboxylated by isocitrate dehydrogenase, producing A-ketoglutarate, NADH, and the first molecule of CO2. A-ketoglutarate is further oxidized by the A-ketoglutarate dehydrogenase complex to form succinyl-CoA, another NADH, and the second CO2. The next step involves the conversion of succinyl-CoA to succinate by succinyl-CoA synthetase, producing GTP or ATP, depending on the tissue. Succinate is then oxidized to fumarate by succinate dehydrogenase, generating FADH2. Fumarate is hydrated to malate by fumarase, and malate is finally oxidized to regenerate oxaloacetate by malate dehydrogenase, yielding one more NADH. Each turn of the cycle produces 3 NADH, 1 FADH2, 1 GTP, or ATP, 2 CO2. The cycle runs twice per glucose molecule, as each glucose generates 2 acetyl-CoA. It's not just for energy. TCA intermediates are also used in amino acid, glucose, and lipid synthesis. Understanding the TCA cycle is foundational for grasping how energy metabolism supports all cellular function, especially in high-demand organs like the brain and heart. Follow for more and learn.